This video covers the IGCSE Biology Objective 3.2.5 on the 2025 CAIE syllabus to investigate and describe the effects on plant tissues of immersing them in solutions of different concentrations. So this is a practical that you may or may not do in the lab. And in this practical, you would have an experimental design where you would have a situation with several, maybe 6 different beakers filled with 100 milliliters of water, and then you would add sucrose in different amounts, which are indicated here. So there is 0 grams of sucrose in the first one, 1 gram of sucrose, 2, 3, 4, and 5 grams of sucrose mixed into the 100 milliliters of water in the beaker. So what we're going to see next is the process of the experiment and then what we would do with those particular results. Okay, so in this experiment for osmosis, you will need to get some potatoes and you will need to use what's called a potato corer. Now, if you don't have a potato corer, you could always cut the potato into slim rectangular sized pieces that could fit into the beakers and still do this experiment. The other thing you will need are 6 beakers that would hold about 100 milliliters of water and some sugar. So what you're going to do is you're going to core the potatoes. And if you were doing this, let's say for a science experiment, as in a practical, this would be done for you. All of this preparation would actually be done for you, but I'm just showing you how it's done. So basically you would just core multiple pieces, and since we have 6 beakers and we're going to replicate the experiment, you will need to have 18 cores, and what I'm just going to do is pop the core out of here, and then I'm going to continue doing this. So I have similar diameter pieces of potato. So let me get that done and then I will show you the next step. Okay, so I've got a few of my potato pieces done here, and what I'm going to do is I'm going to cut them so they are all the same length. So the length that I'm going to cut them is 4 centimeters. 1, 2, 3, 4. And I'm doing this so that I have a control variable with the size of the potato. So all the potato pieces will have this same diameter, which doesn't really matter, but I will measure it, and that is 1/2 centimeter in diameter and 4 centimeters in length. So I'm going to go ahead and do the same thing for all of the 18 potato pieces. Now, one of the things that I managed to do was to cut all of these from the same potato. So that now is another control variable. I don't have to worry about the difference between between different types of potatoes as far as the age or the amount of water that's in the actual potato or the amount of starch that's in the potato or the other different mineral ions that are in the potatoes. Everything will be the same because they're all from the same potato. So that is a control variable. That would be hard to do if you are in the science lab doing this as a practical, because you don't know where exactly the potatoes came from that you are using. But we've managed to do that with this. So now what's going to happen is I'm going to take the potato pieces and I'm going to do 3 at one time, like I've cut here, and I'm going to put them into a beaker of water. The beaker will have 100 milliliters of water, and then it will also have varying solutions of sugar, so that we can see different effects of osmosis based on the concentration in the beaker. So let me get that set up and I'll show it to you. Okay, so I've got my 6 beakers. All of them have water from the same source, all the same temperature. I've got my 3 4 centimeter long potato pieces in each of the beakers. This one has 0 grams of sugar. This has 1 gram, 2, 3, 4, 5, and I'll show you a still picture because I've written the number on the beaker. So now these are going to sit on the side in the same room at the same temperature for about an hour, and then we will remove them from the water and see what the results are as far as osmosis. Will water move into the potatoes or will water leave the potatoes? So that's what we're going to be looking for. So in this particular case, we have changed the amount of sugar in each of the beakers, so the amount of sugar is our independent variable. The control variables are the amount of water, the size of the potato pieces, the age of the potato, the mineral and water content of the potato, the type of sugar, all came from the same bag. So I've tried to maintain as many control variables as possible, so the only different thing here is the mass of the sugar that is in each of the beakers. Okay, so it's been a couple hours. I have pulled the potato pieces out of the water and dried them. I'm not weighing them. We're just going to measure. So I've zoomed in on my ruler. So the first piece is 4.3 centimeters. This is still 0 grams of sugar. Second one is 4.25 centimeters, and the third one is 4.25. The diameter has expanded to 0.6. Originally it was 0.5. All right, so let's go to the 1 gram of sugar. Okay, on to 4 grams of sugar. First thing I want to do is measure the diameter of this one. We got a diameter that has reduced to about 0.45. Remember, it was 0.5 to begin with. All right, so those are the measurements, and what we'll do is we will take the mean of all of those, but before we do the math and do the graphing for the outcome, let's just move that so they're in. I want to compare the pieces that were in the pure water versus the pieces that were in the most concentrated solution here. So we can see the size difference for sure. Let me line them up side by side. Okay, so the diameter of these have increased, and the diameter of these has reduced. The length has increased, the length has reduced. The other thing is that this particular piece is extremely firm. If I try to bend it, it will break. It's extremely what we call turgid, whereas this, if I bend it, is extremely what we call flaccid. And all of them are the same. You can even see the color is a little bit darker whereas the color here appears to be a little bit lighter. I don't know if you can see it under this light, but this is a bit yellowish, and this one is a bit paler in color. So those are the differences. So the next thing we're going to do is calculate the means and do some graphing to come up with our results. So now we're going to take our results to find the actual concentration of sugar in the potato that we used themselves. So first we create our little data table, and so we'll put all the measurements into the data table for each of the beakers, and we'll do that by identifying the change in length for each one. All of the potatoes started at 4 centimeters, and then the measurements are here. I didn't do all of them in the video because I was going through really fast, but you can see the change in lengths. Some of them increased in length and some of them decreased in length. So the first thing we want to do is calculate the average change in length. This is going to be a percent. So we do that first by calculating the mean change in length. So I take my 3 changes in length, add them together, and divide by 3. That's how we calculate the mean. And then we're going to take that answer, whatever the mean change in length was, divide it by our original length, which was 4 in this case, and then multiply by 100. And so then you can see that we're getting these average changes in percentage. Now some of them are positives and some of them are negatives. So we're going to have to create a graph that accommodates this. Okay, so here is the graph where we're going to graph the average change in length on the y-axis, and we're going to graph the sugar concentration on the x-axis. And we plot the points from the data table, and we draw a line of best fit through all of those points. And you can see here that's what that would look like. A line of best fit is we take our ruler and we just draw one straight line. This does not connect the dots. This is an actual straight line. So now what this tells us is that where the line crosses 0% change in length, so where the potatoes themselves, their length would absolutely not change, that length is the concentration of sugar in the potato. In this particular case, that is 3.5 grams per 100 milliliters. So what does this actually mean? Well, we draw our line of best fit, and where the line crosses the 0% change in length, so where the potatoes did not change at all, there was no increase, there was no decrease in length, that now tells us that is the concentration of sugar in the potato. Why is that? Because the sugar and the solution it was in were at equal concentrations, so water doesn't move. Why doesn't water move? Because there is no high water potential or low water potential situation. The water potential is equal because the concentration of sugar in the potatoes also is equal. So in this particular type of experiment, we would do this in order to determine, one of the things that we would determine is the sugar concentration in the potatoes using our data and this type of graph.